Thanks. We're going to go ahead and get started, folks. Alrighty. So what you guys are seeing right now is this is real-time nanopore sequencing. Right now, it is going through. It has gone through 2 cycles of MUX. Now, what that MUX means, that's the selection of active groups. So as we discussed, in each channel, there are 4 proteins that can collect data from at a given time. It's going through each channel, each pore, and deciding, is this a good protein or not? Is this a good pore? It's passing voltage through it, measuring this voltage, measuring characteristics of what it thinks is a good pore, and deciding which group it belongs in. There's 4 total groups. Now that's what this panel here is showing. This is just your log here showing what each step it's going. So we're on MUX 3 now. There'll be a total of 4 MUXes. This here is the pore panel. This gives you real-time information about what's actually going on with the individual pores. Now, there's a number of different keys. Some of them are fairly esoteric, unfortunately. But the black is saturated. Those are channels that they're not able to collect any data off of at all. So that's, nothing's good. Nothing's passing through. You're not going to get any data off of that. Infinity to 0 picoamps. This is not commonly seen except when they intentionally do it. This is just measuring what if the picoamps are in a range they expect it to be in. 0 here, which are these guys here, are more or less similar to the saturated values. There's information coming off of it, but as far as I can tell, there's no DNA in that pore. The pore isn't working. There's something wrong with the pore. This dark blue one is picoamps to positive infinity. Again, this is a measure about what the picoamps are for that particular pore in that channel. Now we're in the more interesting pieces. Single pore. This means the pore is good. It is functioning, but there's no DNA in that pore right now. Great, okay, that's not a problem. Strand means it's there, it's sequencing, but there is DNA in that pore right now. Okay, now this is going through its cycle. This is starting MUX 4 now. Multiple means it's getting information from more than one protein at a time. There's crosstalk between the proteins, or there's multiple pieces of DNA in there, but something makes the signal bad. So this is also a bad thing, and then unavailable is yet another bad value. I'm not quite sure how unavailable differentiates from other portions, but again, that's not what you want. So it's cycling through, changing the voltage. It's going to change the voltage positive and negative throughout the course of the run. It's testing everything. Now, one of the unfortunate things I'm seeing, which happens, is that there's not a lot going into strand. The flow cell is good because single pore is very high. In any one group, you're not going to see 512 single pores. That's fine. But I'm seeing not much in strand, just a few pieces of DNA. Now, this is very possibly due to the fact that in this run, the average size of DNA is about 550 base pairs long. So the nanograms of DNA material present is very low, although the molar concentration is similar to what we would expect from a larger fragment system. So chances are the diffusion, the movement, the ability of the tether to capture the DNA is lower than is ideal. And so that's only capturing a few strands at a time, which is what kind of we expected. So that's fine. So we're going to wait for it to finish its process, then I'll kind of show you what the group segregation looks like. Let's see here. Now, there are other values that can be seen. I don't think I can easily access them. But you have all this information up here. This is going to give you information of how long the run has gone, what the current yield is in events, name of the sample, the data sample, and this is assigned by Oxford Nanopore, your ASIC temperature, okay? The ASIC is a particular temperature, that's the actual piece that's inside the flow cell, and the heat sink is an instrument within the flow cell, the instrument that's pulling heat away from the ASIC. We don't want that ASIC heat too high, you have protein denaturation, and that doesn't work. Bias voltage, there's different things that you can manipulate, which, let's see if I can't get to those other values. Here we go. We got some other information here about what's going on, voltages, temperatures, controls. Some of these values can actually change, but let's see here. Unfortunately, we're on a screen that's making it less than easy to work with. There you are. There's the message viewer. Okay, so here is our groups. Now, unfortunately, it looks like the number of groups available has gone down. This is last time I QC'd this flow. So this flow cell initially QC'd at 460 pores in group 1. It's now at 379. So this is group 1, okay? Then 177 in group 2, 59 in group 3, and 8 in group 4. Now, generally, you're never going to see any data collected from group 3 or 4 because the software currently only goes from group 1 for the first 24 hours, then to group 4 for the next 24 hours. Sorry, not group 4, group 2 for the next 24 hours. There are scripts, there are ways to modify that selection that you can change that, but typically the ones in the later groups just aren't as good. What we hope to see, yes?
So that means that essentially 50% of the chip is not even used to read any data?
Yes.
Okay.
Now, what we hope to see usually is this value is over 450, and this value here is actually hopefully pretty darn close also to 450. The big jump here we see, that indicates to me that this flow cell is probably not going to perform up to what I would think is good, which is unfortunate, but you have to kind of deal with what you have. So you have to, with a new technology as this one, you have to kind of be willing to accept that sometimes things are not going to work as well as you would like, especially when trying out a new protocol. Okay, so let's shrink this guy down. Let's bring this one up. Okay, so here's what we see here. Now, as you can see, the strand numbers are slowly going up. That's really good. That's what makes us happy. That means we're having more of these DNA fragments diffusing down to the surface of the flow cell and then getting captured by the tether and brought to the surface of the flow cell. There's still a fair number of things in single pore. Now, when you see this situation, that probably means you really haven't saturated your flow cell. You can afford to put more stuff on your flow cell and still expect sequencing to happen. So ideally, we want to see many, many more things in strand than in single pore. Fortunately, over the course of the run, we'll see more and more of those strand values increase as the flow cell runs. So we're at 34, 36 now. Single pore decreases. Now here, we can look at what each of the pores are actually doing, okay? So we've got three different pieces of good information here. Here, this is the squiggles. This is the value from each of those selected proteins, and this voltage is actually being generated by them. This is the size fragments of the sequences that are actually generated. Now, if you'll note, I have most of my reads in this 1,000 base pair bin, 1,000 to 2,000. That is good. That's what I expect. This library that I prepared, the average range is from 400 to 750 base pairs. So this is what I expect to happen. This is real-time feedback of library performance. Now, if I were to use a larger library, what I would see is I would see some fragments coming earlier than others. So early on in the run, there is just based on diffusion, smaller fragments tend to be captured and sequenced first, and larger fragments tend to be sequenced later, partially because it doesn't deliver that fragment size until after the DNA is fully translocated through the pore. It takes longer for a longer piece to translocate, so they don't show up over here in these ranges. Now, I will see a few of these fragments that are in excess of what I expect. Those could be one of two things. They could be simply pieces of DNA that weren't fragmented to the size I expect them to be, or they could be artifacts. Sometimes the DNA gets stuck in a pore, or you have anomalous signals, or the pore gets confused, and you end up with these reads that are very, very, very long, but you see they're like 500 A's in a row. So that's not what you want. So these may be artifacts, they may be real. This is the number of channels that have actually generated a read. We want the value to be up here. This will grow over the course of the run. Right now, we're at 217. That's half of them, like you mentioned, are actually producing data. We're hoping that it will increase as the run goes on. Complete reads, this is when it's done, okay? And events in read, so that's a 6-mer in this case. So this is the number of 6-mers that have been identified. It is a rough, and it's very, very rough estimate of how many bases have gone through this sequencer. You don't divide it by 6 because there's one event per base approximately. But this gives you a very general idea of how many gigabases you've actually sequenced. So right now we have sequenced around 600,000 bases. So not too bad. This is all real-time information. All right, now we have more information that we can go with. So this is an interactive panel. We can do different things with this panel, and you can't see those, so let's pick a different pore. There we go. All right. So there are these different options that we can do. So let's hit show on that pore. Nope, that's not the one I want. Let's see here. Show hide range. Show channel to channel 1. Okay, that should work. Why aren't you? There we go. Okay, so what I've done is I've set it so that I'm only showing this squiggle for one single pore. This lets me look and assess what's going on in squiggle space at that given time. And unfortunately, I don't have access to this portion of the window here through this interface, but you can actually change this window, and you can zoom this in and look closer at the squiggle as it's actually coming out of the pore. I can also look at groups. Now these are organized based on the way the device is oriented into groups of 8. The way it's machined, there's groups of 8 on there. And so you can actually set to looking at groups of 8 of the voltage as it's passing through the pore. And so you can look at good pores, bad pores, yellow pores, green pores, and see what's going on in each of those individual ones and investigate if things are performing the way you expect them to. Now, if you want to be particularly handy, you can also say, take a pore here, and you can change features of that pore. So right now, if you look at these different pores, there are little black triangles. There's one there. There's one, let's see, green triangle there, green triangle, black triangle. Those are very hard to see, but they tell you which protein within the group of 4 is being used to collect data. So this is 1, 2, 3, and 4. Okay, so group 1 are all the good proteins, but there's 4 proteins within a channel, and one of those 4 proteins is group 1. So I can actually change if I desire. Let's find one that I don't think is going to mess things up. Okay, this guy here, this is, I believe, in group 3. I'm going to change this guy to go to group 1. Now see how the triangle changed from there to there? Now I've selected manually which protein I want data to come from. Now, it looks like, as you can see, now that pore is turned to light blue. So that choice of protein 1 was a bad choice. Now I'm not getting anything off of this pore. So I'm just going to go and switch it on back to the way it was. Alrighty. I can do it for everything. Don't necessarily do that, but you can say, all the pores, I want you from protein 1, all from protein 3, all from protein 4. I haven't found much utility in that unless I'm actually playing with it and trying to optimize stuff. It's worked a little bit for optimization purposes to set everything to one channel towards the end of a run, but generally you don't want to do all that. Mux open, that's just going to open the pore. It's going to let whatever you want to go through it. There's no it's total voltage passing through. It's not going to collect any data from you. I actually don't know what mux test current open pore is. I haven't figured out what that one does. I've run it a couple times. It doesn't seem to do anything for me. Mux unblock voltage. So as DNA goes through it, sometimes it seems to get stuck, and sometimes you want to reverse the polarity across a pore to free the DNA that's stuck in the pore. So if you think that you have DNA or material that's stuck in a pore, you can actually reverse the voltage on that particular protein, try to free it up, and then continue to get sequencing information out of it. And these mux test currents, these are things that I don't ever actually use. Mux ground, that's actually going to be these black ones. That's going to say, okay, off, don't collect anything data, no voltage, nothing. I don't know what mux ground through resistor is. So some of these things are things that Oxford Nanopore themselves have it programmed in there for their own testing and manipulation purposes. Generally, though, it's those changing the proteins and doing the unblocking that's important for your average researcher. Now, if you see, since I've been talking, our number in strand has increased here up to 50. So we're hoping to increase that. In our experience, it takes about an hour for you to reach actual pore saturation. So the hope is that as the run goes, this number will remain at least constant and not crash. Unfortunately, if we come back tomorrow, you'll probably see this number has gone down fairly drastically. Okay, where's the bias voltage? Here we go. Okay. So there's actually some buttons over here that I can't get to right now. But this bias voltage, this is a bias that's set to pass the voltage through the pore, which maintains a certain characteristics relating to the number of ions on the cis and trans side of the flow cell. As the process progresses, you will see that this bias voltage will increase. So it's applying a greater and greater external voltage across the membrane. This is because the buffer ionic concentration on the cis side of the flow cell and the trans side of the flow cell has changed. So they need to change the voltage to control the way the DNA translocates through the pore to maintain as much as possible a constant value. Let's see here. Data streaming, that's on. That means that there's some portion of this data that's going to Oxford Nanopore for real-time troubleshooting, which you can do. We want to see that the target temperature does not change. You can change this value to something else. It'll give you a warning if our target temperature changes significantly from 37 degrees. Hot is what the problem typically is. It does not control the heat properly. Yes, you want temperature control on. I can't think of any reason why you'd want to turn that off. Test current, I don't change that. Sampling frequency, this, if you want to really get into the data structure and start manipulating what's going on in the most raw sense, this is a value you may want to potentially manipulate. This is the number of times it tests the voltage over the course of a run. Now, the more times you sample, the more computationally intensive things get, the bigger the data sets tend to get. But the fewer times you sample, the less resolution you have. So you want to pick between computational power and the sampling frequency for resolution. Integration capacitor, I believe that has to do with the way the data is visualized. I've never changed this value before. And this is just a report here. Estimated events per base. We want this to be 1. 1 is the perfect one. That means each base has approximately one event, approximate event associated with it. It's 0.9. Okay, which means there's some events that aren't going properly. There are more bases than events, so we're skipping events a little bit. That's not surprising that that happens. And the cumulative events, 2.439 million. Alrighty, what are some other things? You can stop and start protocols. I don't recommend it. Some people have done that in order to reselect the pores. So if you actually stop the protocol completely and then start it again, what you'll see will happen will be that the selection of pores for group 1, group 2, group 3, and group 4, it'll pick a different subset of pores because they've started to denature, the ion has changed, so pores that were good before may not be as good now, but that bias voltage is problematic, okay? So if you restart it, and turn the software off and turn it back on again, the bias voltage is going to be at 140 because that's what's supposed to be at the start of the run. But the appropriate bias voltage, depending on when in the run you started, might be lower than that. It might be need to be 150. It might be need 160. So you're going to see a real decrease in data quality if you stop and restart a run without also changing that bias voltage to reflect what the ionic concentration is in the cis and trans compartments of that flow cell. Okay, let's see. Disk usage, you can't see that. Keep an eye on that value. I have definitely run out of disk space during the course of a run. That'll crash your run. So if you accidentally leave a couple of runs on your computer and you run out of space, then yeah, it's not nice because you're like, this is an awesome run, so much data. Oh man, now you've crashed your run and you're not collecting any data anymore. So that's very frustrating. Make sure you keep an eye on your how much stuff is being generated and delete stuff as necessary. Okay, so the metric core software, what that does, that allows the base calling. So what happens is your reads as they're coming off the flow cell, they are sent off, beamed into outer space to their base calling software, and it's going to start calling the bases on them. It's going to say, okay, A, T, G, C, so on and so forth. And then it downloads them back to you. And it sorts them based on a Phred score in terms of pass and fail. And within the data structure, there's more information. We'll go over the data structure here shortly. But you can monitor how well your run is performing based on the number of reads that are going into that pass and fail folder. Okay. I'm going to shrink this window down, and we're going to pull this guy back up again. Okay, data structure. So as your data is being generated, you're writing to your disk FAST5 files. Now, these are frustrating data structures. There are other ones out there. Some people say these are great. Some people say these are not great. This is a nested data structure for large data, metadata collection, and value collection, which you have different types of characteristics and features. I'm sure someone out there knows much more about this data structure than I do. I find it tedious to manipulate and get through, but it does contain a lot of interesting data. Unfortunately, Oxford and Nanopore has this very nasty habit of changing their data structure on a week-to-week basis. So all the tools and analyses and whatnot that are there for extracting data from Oxford and Nanopore crash every few weeks because they've changed the data structure and now it doesn't work anymore. Fortunately, most of the people who are building tools are very, very good at being very quick on fixing the tools to account for the new data structure. But be warned, if you do start using Nanopore data and some of the tools you're using suddenly don't work, that's probably because Oxford has changed the data structure for new reads. Now within the FAST5 file, there's lots of interesting information. One thing you get is the least processed piece of data, and this is going to be your events. Okay? You have an event approximately for every base, and this consists of several values. You have the mean value, you have your standard deviation, your start and Unix epoch time, and your length. That's the duration of the event happening. So you've got about the dwell time lasting for longer. This is information that will be used in the hidden Markov model with the verbidity algorithm to determine what the identity of that base is. Now, we have a higher level of complexity. This is still event space. This is base called events. So this is going to share many of the same values you see in the event data set, but we're also going to include things like the predicted 5mer or 6mer, depending on whichever you're looking at. You get this, it's called the level in this thing, because the level is the state that it's coming from. Sorry, states, not level. I apologize. The level is the expected value. The state is the 5mer. You have move. Okay, move. That's an interesting concept. Sometimes events are missed. Sometimes events are doubled. So the move is saying, okay, this event means I've shifted my 5mers down one base, 2 bases, or no bases. So because this is the first called 5mer, the move is 0. We would expect each additional step to be 1. Because that means it's moved 1 base based on what they predict this 5mer is and what the previous 5mer was predicted to be. If you see it 0, then maybe it's dwelling longer. Maybe you're seeing a weird polymeric sequence. This is going to be true for your homopolymers. If you see 2 or 3, then you've missed events in there. So that's used in some of the more sophisticated methods of aligning this sorts of data. We have the weight, which is, which value there, right under the move, this guy here, 0.999995, okay? That's one, that is partially because this is the first base, but there's also weights associated with each of the possible 5mers. If you recall from their verbidity algorithm, there's only 4 possible expected states following the appropriate path from one 5mer to the next. So those 4 possible states have a much higher probability of the choice of move than other states. We don't want to totally exclude the possibility of other states because we've already said that we can skip events. We want to decrease the probability of those events from happening. So that's what that weight value is there for. And then we start getting some of the probabilities of the actual function, okay? The probability of the model state, probability of the pico, I can't remember what the MP stands for. Probability is A, C's, T's, and G's. These are all values that go into the model. Now, I cannot tell you how these numbers are used to build the model. Machine learning people out there who know hidden Markovs, please have fun. But these are all values that are used for predicting this nature of that 5mer, whether the 5mer is correct, and how far it's moved. And this exists for both complement and template, okay? So each strand has its own associated characteristic values, okay? So it's moving in a different, the DNA is moving at a different speed, there's different voltage, there's different subtle changes in the way we expect the values to be as each 5mer or 6mer is in the pore. So we have variations in the values. So if you look here, this is for poly A. the mean model value. So this is what they think it should be, not the actual events. The mean model value for poly A is 70.5948. That's for the complement. I believe that's complement strand, okay? And then for the template, 70.2417. Now that's not very big. They're very similar, but still important. And you'll see that's true, okay? Some are more significant. This is 68, this is 66. They'll use those based on, after they've deconvoluted which strand they're on, to model and decide what the 5-mer is that's associated with that particular event space based on all the parameters collected. And there is standard deviations, means, weights, they're all associated with those particular values for the model. These are all also used as a method of determining what the 5-mer is. Now, changes in the chemistry, which happen often, not as often as they used to, necessitate changes in the model. So you can't use these values on, say, map 0004 chemistry. Right now, we're on map 0006 chemistry. Okay, so there's slight differences in the concentration of the buffers, in the way the flow cell is constructed, the nature of the proteins, different things like this that are all going to necessitate slight changes to the model. So if you're an algorithm person and you're working with this data, make sure you're keeping up to date on what the current model state is. There are, of course, also more values that go into the model. I don't know what most of these are. For example, drift refers to the expected variation of the mean voltage over the course of a run. So even though we are changing the bias voltage to try to account for that, different bases exhibit more drift and more changes in their expected event space. So they try to model that as well. So every possible thing that can be measured for this stuff is modeled in this. And if you dig through these FAST5 files, which I find very tedious, but it's all there, you can get every value that they're using in their really complicated model for predicting those 5-mers. And that's because there's not a lot of difference between each 5-mer. It's very, very subtle. So they have to collect lots of information to try to model those minor differences to accurately predict what each 5-mer is. Okay. Back to the question about the hairpin. The hairpin alignment. This is just one of several fields that associates the hairpin. There are characteristic bases, there's abasic sites, there's certain sequences all associated with the hairpin that when the system goes, hey, I see these characteristic voltage patterns, that must be the hairpin, okay? And those are all recorded also in that FAST5 data structure. When it identifies those and sees those, then it switches to the complement model for predicting the base set. I have seen instances, however, where the model messes up, and if you go to the event space for a really long 1D read, you'll find characteristic patterns that you manually can say, that must have been the hairpin, because it's not always perfect. But these characteristic voltages, these positions, these alignments, that tells the system where you are in the sequence so you can predict which model to use for calling which 5-mer or 6-mer base sequence. I think the hairpin's actually on the protein. So there's a protein that's associated with the hairpin. So I think the his tag's actually on the protein, but I'm not 100% clear where it's attached.
That's not what I'm looking for.
No, no. It's abasic sites and sequencing that they're looking for in the hairpin. Okay. Now, I've got template, I've got complement, I've got predicted events. There's another layer of modeling on top of that. So I've got a forward and a reverse value for each strand, hopefully. Okay, now those should be complementary to each other. We should know what each piece is. So now the algorithm during base calling, okay, will take those pieces and go, okay, I know this one goes here in order for the template and complement. Then in K-mer space, not full read space, they try to align the template and complement strand, okay? So what you have here is you have the predicted 5-mer and the position in template and the position in complement, okay? So we expect that this value should be found in position 0 in the template strand and position 929 in the complement strand. And you'll see it iterates ding, 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 as each one goes up, okay? It doesn't always work well, but if you look here, you can see that shift. So this is T, G, T, C, T, and then we have G, T, C, T, G. So you see how they line up. They shifted over by one base. This and this, they're the same. You have a problem. That's reflected also in this -1 value. This -1 value means the model has no where that piece goes. It's confused. So when you see this -1 shifts, in the position in one of the strands, that means, okay, we don't think this fits right, let's not use this. And they will try to account for those values when they produce the consensus sequence. Yay, something actually useful comes out for those people who are not algorithm people. Within the base called final processed FAST5 file, you get the complement and template sequence and the 2D sequence. There's 3 sequences that you can extract from FAST5, as well as an associated quality for each template, complement, and 2D, which is very useful, okay? This is the real stuff that you want to look at unless you're trying to work on algorithms. Now, you guys don't want to go through and manually pour through FAST5 files and extract this information. So the wonderful people who've been involved in that program have been building lots and lots of tools to allow us to manipulate and extract useful information. The very first tool set, and very most frequently used probably tool set, is Pour Tools by Nick Lohman and Aaron Quinlan, who you guys will be meeting later on this week. So if you have questions about Pour Tools, ask Aaron. Within this set, there's a lot of functions that are very, very useful along here. This is a command line based tool set. It allows you to extract the fast A's, the fast Q's, create various different plots for read length, generation, squiggles like here, so you don't necessarily have to look at the interface with a nanopore to look at the squiggle space. You can make plots, find the longest read length, produce things in tabular sequence, so on and so forth. Nice, convenient initial string use for looking at the pools. Now this follower sheet's all wrapped around, so you don't need to worry about seeing where things are. One of the more useful things I found in Pour Tools is it's fast to extract the events. So all those singular blocks of events you saw that are all stacked on each other, not useful for downstream computational approaches. The event extraction tool, that pulls out all those events, puts them in a tabulated format. Now it's in a format that's usually easy to use. Gives you all those probabilities, those models, template, complement, everything you want to know to build your own algorithms. If you don't like command line tools, at least not Unix-based command line tools, if you're an R person, go to Pour. Okay, Pour R is Biomic Watson's paper. This is going to be very, very similar to Pour Tools. Many of the same values and plots are available through the R portal for Pour R as are in Pour Tools. So it's a matter of preference in this case. I like the plot outputs a little nicer in the Pour R than in the Pour Tools, but that's simply aesthetics. One of the more useful plots that is a little bit different than what's in Pour Tools is the yield occupancy values. Now you can get that out of Pour Tools, but the plot I think is very nice. What this says is this tells me how many pieces of DNA each pore is processing in a graphical format. Okay, so the darker it is, the fewer, the redder it is, the more it's processing. So this I think is a very useful visualization of how the flow cell is performing on a pore-to-pore basis. And you can of course also get tabular format for each of these values as well. All right, let's see here. Yes, we have time. So unfortunately, I wanted to do a live demo of Minotaur, which is a very useful piece of software, but Oxford changed the data structure, and the proprietor of Minotaur told me yesterday that he can't actually do a live demo on my live data because it's not compatible until like Monday. So I'm just going to show the video of Minotaur to get an outline of what this does. This takes in the stuff you saw in Pour R and Pour Tools and makes it a more unified analysis pipeline.
It's the end of the year. I decided to record a quick video to show you the latest version of Minotaur. That quick video turned into a 35-minute long description, and I'll link to that elsewhere. So here is a 5-minute video describing the latest version of Minotaur. So Minotaur has undergone a few updates. If we have a look at the current sequencing runs here, you can see we're uploading 5 different runs right now. I'm just going to dive into one of them to look at. You select a run by clicking on it, and that immediately directs you to the overview page. There are new things happening in the background now. We're analysing data on the server, and so you can see this read, upload and processing window, which is new. It shows you the number of files uploaded for 2D complement and template, the number of reads that have actually been called for each of those, and then the number of reads which have been processed and aligned. So they're smaller fractions. You get your read and coverage summary in various different forms. We now break out that alignment to the reference by each of the files in the reference, and you can see that we have the rates of base calling, number of reads per pore, and also now a view which shows you the layout on the chip. This doesn't show the numbers of reads, it just shows the physical layout. The intensity of the colour shows you how many reads are being generated. So this actually reflects the layout of pores on the chip. We show the length plots and various other bits of data. You can go in as well under the data summary view here and see various new features, one of which is the read histograms. So we're showing the number of reads both by read length and the number of bases sequenced by read length, and you can filter out different subsets of the reads and just look at the ones that you want to here. Clicking on any one of these bars will open a new file, FASTA file, containing the reads of that length. You can do all of this in real time. We have rates of sequencing, various different plots. We have pore activity, so this is the same sort of thing that we've seen on the front. Read qualities, these take a little bit of time to generate. So here you can see them, and okay, I've cheated a bit there by cutting out some of the information, but you can see the number of reads we have of each length for both the template, complement and 2D, and read quality scores for 100 random reads. And the nice thing you can see here is that the 2D qualities are on average higher than the complement or template. We then have coverage detail plots, which are plotted for each reference, so you can see those here. And we also plot where the 5' and 3' reads are mapping to, the 5' and 3' end of each read are mapping to. Then we have some new plots here. This one is slow, and I will edit the video to compensate for that. So this shows you the bases at every position. What we do is we take the alignment and we look at the base that has been called for each sequence where it maps to the reference, and we then score that and log it. So we've got the template, the complement and the 2D. I'm just going to zoom in on a tiny fraction of this so we can see it in a little bit more detail. So here we are zoomed in. Now hopefully you can see that the colours correspond to bases. So blue is A, yellow is T, green is G and C is red. We then also count deletions and insertions at each position. So you can view exactly what's happening, and we'll come back to this plot in a moment. This is under development. We're looking at it in more detail as we go through. So we also have this WIM plot, and this essentially shows you the cumulative number of reads over time for 3 different reference sequences. In this case, this is amplicon data we're looking at. You can see that 2 of these are particularly common, one of them very few reads are mapping to. This plot is obviously a lot better if you're thinking about lots of sequences which are very divergent from one another, but essentially it shows you what's in your minion. So that's the data summary. Individual read data, you can go in, you can find reads, you can sort reads based on length. So I'll sort these by template length. It might be more useful to sort these by 2D length, because that's more reflective of what we're looking at, and also you can see that these reads align to the reference, so that's nice. So if we just take this top read, we now get shown some data around that read, and that data is the squiggle plots for the process read, it's the quality scores, it's the base probabilities, and it's the bases that have been called themselves. And you can zoom in on this, you can zoom out a bit, you can browse around and look at different parts of the trace as you see fit. You get the same thing for the complement as well. You can also change the order. This data is being presented in time order. If you want to see it in base order, i.e. just the order of the bases removing the time element, I'll pick a shorter read for this one. You just tick the base option and it now generates that data in base order. So you can do some interesting things just looking at individual reads here, seeing if particular positions are potentially variant, show different things. If you scroll down, you get some information about when the read was called, and you also can download the template, complement or 2D sequence from that read in either FASTA or FASTQ format. You can export the entire read set at this moment in time, download fast A, download fast Q. We also have a new feature which is a run report. This gives you all the metrics from Minno, Metricore, the reference that you're using, various Minotaur reports, and you can also have a live commenting system to keep track of what's happening and to add things onto it. This is maintained over the history of a run. Even if you re-upload that run, that report remains. We still have our alerts for coverage depth, for base notification. We haven't really worked on this much, so it's something for the future.
Yes, you're midnight.
The newest change is this nucleotide variant plot, where you can have a look at your data and look to see whether there are any particularly variant positions. We look at consensus variants, we look at sequences which are variant more than the average, positions that are variant more than the average throughout the data. Let's choose a variant position here. I'm going to sort these by the total number of bases called to get a really nice one. Here's one here. It should be an A at this position. It's in fact reporting a G as the most commonly called base. If we click, we can zoom in on this. You can see the reference sequences plotted in the lower panel. Where it's 0.5, that means that the reference matches to the sequence that's been called. Where it's 1, it differs, so let's zoom in on this. Here is the different position, 9065, and indeed you can see a G is being called more frequently than the A, and that this differs from most of the sequences around it. You've got various options here. You can choose to look at template complement or 2D. We say 2D by default. You can also select the minimum coverage for looking at these variant events. These aren't variants, these are just events which are variant. You do the same thing for regions which look unusually variant. So here we have, for example, a C. It's the consensus is being called as a C, but actually G is being called quite frequently. This is at position 115, so let's zoom in and look at position 115 in more detail. 115 is here, and here you can see the C is being called, but the G is quite common as well. What you can do is switch off different bases from these reports and have a look at what's happening. So let's remove the deletions and the insertions. Let's just look here at G and C to see where they're being called, and we can see that G and C are being called reasonably uniformly. Let's just compare it to the reference. Let's hide the A's and T's. So there might be something you can learn from this when thinking about these variant positions. We also look at deletions where they're more common than average, and insertions where they are more common than average. That's a very quick summary of the current sequencing. You can export reads and do all those things. Previous runs, you have access to exactly the same features. We can change a run here. Let's look at an old Lambda run. Here it is. You can see the data summary. You've got all the same plots. You've got individual read data, exporting reads, run reports. The nucleotide variants, the only difference is here you can actually export the variants in a CSV file if you wanted to look at those further. That's a really quick overview of Minotaur. If you want the longer version, it'll be linked to from this post.
Alrighty. That was Minotaur. So you've seen a R interface, a Unix interface, and a GUI interface for doing much of your primary analysis on your Nanopore reads. Here we go. So there is another one. This is Nanook. This is yet another thing similar to Nanopore. Minotaur. This is not going to have quite the same level of alerts and interactivity, but it's a very similar process as well. Nanook, Nanook of the North, polar bears, that's, what the folks named it. It's a flexible multi-reference software for pre- and post-alignment analysis of nanopore sequencing data, quality and error profiles. Has general functions similar to that used in Pore tools. Extract, Pore tool, extracting the FASTA, aligns against the reference, typically using BD2A or LAST. Also does Blazor and margin align, and we'll touch upon margin align here in a minute. Generates a wide variety of reports. It starts from the FAST5 files, you feed it into the FASTA, and then it is the alignment for you and produces a suite of reports and graphs in a PDF format. With something that is not implemented in Minotaur is the output PDF format. So you can generate all these different comprehensive GUI type reports in this GUI system. Individual graphs, tab separated files. Homopolymers references abundance. This is not something that Minotaur does currently. What you can do is you can see if each particular 5-mer or 6-mer is over or under abundant in your sample, which generally we'll find that they will be. Some values are more divergent than others. So if you look at template here, the template is much more divergent in expected versus actual abundance of 5-mers, and the 2Ds, because the consensus read, is much more accurate. Lots of report options available, error analysis, read lengths. You can all set and tune what reports you want to see in this PDF. Alignments, lambda perfect k-mers. This is going to be just k-mers. You're going to see how many of these perfect runs, longest perfect sequence you're going to generate. Coverage profiles. X-mer analyses, if you want to see the overabundance of particular 5-mer, 3-mer, 10-mers, whatever, you can get that to outgo. Motif analysis, and it's in process right now. So similar function to Minotaur, produces PDF values, analysis, general structure of your fast 5 runs. Okay, next level of difficulty. You've got your data, you've QC'd it, you've seen your 5-mers, you've seen it's good. What do you do with it now? You want to assemble it perhaps. Well, we've already discussed that Nanopore data is fairly errorful, okay? So there are several different approaches out there for correcting errors. One is the Nanocore platform. The Nanocore platform is going to be using MySeq reads to correct the Nanopore reads. As you can see, fairly high error correction. So before correction, we have a percent identity here on this axis and the read length. And if you look here, the percent identity is about average 72 or so, okay? And this is true across all the calculated read lengths, okay? We also find that a lot of those reads that are larger than 60,000 don't align. Again, that's because it's very, very error prone, very difficult to align to those very long reads, and there's very few of them. All right, so how does it work? It starts with MySeq correction. Step 1 is a lenient blast alignment to MySeq reads, and by lenient I mean we don't have a lot of penalties, we allow gaps, we allow mismatches. We don't want perfect alignments because you're not going to align hardly anything if you're very, very stringent. We next sequence the reads with an LIS algorithm to maximize length of full reads and minimize the overlaps. This is going to help us find the best choice of reads to go into the final value. It's next built with a consensus, so we're stacking those reads that have been filtered on top of each other, finding the probabilistic path through them that minimizes the number of errors through the reads. And finally, those are assembled with the Celera assembler. Now, the Celera assembler is something that was used by PacBio. It's much more efficient for long reads than some other assembly algorithms. So you'll see that most of the Oxford Nanopore stuff uses the Celera assembler to assemble. So after the Nanocore pipeline, we see the percent identity has now exceeded 90% on average, and we're still seeing a number of reads in excess of 10,000 bases. So works very, very well if you want to use MySeq hybrid error correction approach. If you don't, then you move to this suite of tools. This suite of tools is NanoPolish, NanoCorrect, and Event Align. This is in one of the Nick Lohman's papers here. All right, so those three pipelines occur at different points within the process. Input reads is your fast5 files. So I'll touch event align momentarily. Error correction is where we're at right now. Their NanoCorrect pipeline that is going to fix those errors in the Nanopore reads before going on to a Celera and then consensus sequence and then genome sequencing. This is a little bit different than the PB consensus for the Nanocore. We'll touch on that because that has to do with the NanoPolish tool. All right, NanoCorrect, partial order graphs. They're laying the reads on top of each other, finding out where they deviate, okay? Each one of these is lines between the nodes, the number of reads that match this value. So this maybe has 10 reads that have a C in this position and T in this position. This maybe has 8 reads with a T in this position and a C in this position, and 1 read with an A in this position. Then, again, these are things that people who do algorithms will know better than I do, but then they find the best, the most efficient path through the data to find the consensus and the corrected value to go into downstream applications. This is Oxford Nanopore only sequencing, okay? So there's no MySeq used for this error correction approach. NanoPolish, now we have the hidden Markov for calling the 5-mer bases. This is a hidden Markov on the original hidden Markov. I'm just tickled by this concept. What nanopolish does is it attempts to model the expected errors from Oxford Nanopore. So they already know that there's certain types of errors, indels, insertions, deletions, skips, event jumps, whatnot. So they are modeling what they expect to see in the error space, and then fixing the Oxford Nanopore reads based on what they expect the errors to be. So they use a hidden Markov model, lots of probabilities, lots of complicated math. My husband tells me there's apparently terms missing on here. So I'm not sure this is actually accurately represents what the real model is, but anyway, they're predicting the states matching event to the 5-mers, the probability of extra events, the probability of missing events, based on how they understand the Oxford Nanopore error profile to work. What they argue is that you can have a significant increase in accuracy of assembly. So this is their draft before the polishing that's already assembled, okay, before the nanopolish is applied. We want everything to be on this axis here. 5-mer count is how they're expressing efficiency. There's still these 5-mers that are off target here. This is after their nanopolish hidden Markov algorithm is applied. We still have a couple guys, homopolymers, that are off axis, but we see an increase in accuracy in terms of the 5-mer proportions in those two different approaches. All right, event align is something that's fairly new. This is going back to even more raw data than before. All of the alignments more or less rely on the consensus sequence, corrected frequently, but these are full length 900, 2000, 10,000 base pair reads. However, event align takes the k-mers, these 5-mers, these 6-mers, and goes, okay, I don't need to rely on what Oxford says the actual read is. I'm going to go back to a more unprocessed level because I can figure it out better. So the event align takes the k-mers and does a chaining tiling approach to put those k-mers together and align those to a reference. So they're like tiny, tiny, tiny reads, like 6-mer reads, but they use information about the surrounding reads to build them in an appropriate overlaid manner as opposed to, say, using short Illumina reads. And that's event align. And these are all part of the nanopolish suite. Again, these tools are all available on GitHub, so if you want to use these tools, all available on GitHub, easy to find online, but I'm happy to share the websites if you would like me to.
Sorry,
yes?
How does it start with its search? If it's going to start with it's going to search the whole thing with a 5-mer?
It's a very good question. I honestly don't know how they start. I do believe there's some early chaining of events. I don't know what their seed size is. I'm sure it's got to be more than 5. You're absolutely right. This is a recent tool. I haven't gone through all the documentation for it. Okay, margin align. Now, this is a aligner that is dedicated to variant calling. So structural variation, copy numbers, large things, not necessarily all that big of a deal to have a high number of SNPs, a high number of base pair errors. But if you want to call SNPs, you kind of have to go from a different approach. So the margin align tool, this is dedicated to ultimately getting to SNP calling on your Oxford Nanopore data. Now these are the various different tools that are used in the option, and this is margin align. They have a standard aligner, which uses last. Last tends to be the most popular aligner used for Oxford Nanopore data. These are fairly lenient parameters because we want to account for those errors. BWA has included now their own tuned version for processing Oxford Nanopore. The most recent version, I can't remember which one it is, but it's got that 2D parameter to use. You can set it to do in chaining or non-chaining approaches, but this EM mode is what we're going to focus on. This is the significant difference in margin align versus some of the other tools. This is an expectation maximization function. So it's going to take the alignments from these guys and make them better. All right. So what they're showing here in this paper is they're showing that the tuned values, those are these squares, those are using parameters in those aligners that are, the dots, those are the tuned ones, parameters in the aligners that are recommended for Oxford Nanopore. The squares are the default alignment parameters for the different tools. Default doesn't work real well, but we knew that. The dots, those are tuned. Those are not gone through expectation maximization. Those are the initial alignments that it does. Improve the performance, and then we see the expectation maximization, the EM polished values increase in accuracy. There is a bit of a toss-up. You do increase your indel rate slightly with the EM. It's going to improve your mismatches. That's important because you're sort of calling SNPs, but you do sacrifice some accuracy on indels when you're doing the EM function. They do see some substitutions are more common than others. This is why they want to run this. Oftentimes you see A's and T's and T's and A's being substituted for each other. G's and C's, a little bit different, but sorry, these are, I apologize. The G's and C's are the most likely to be substituted. The A's and T's least likely. Alrighty. You can get the alignment stats, how many deletions per read base, how many insertions, how many mismatches, coverage, these sorts of things that are generated through many different tools, but specifically aimed towards the sort of metrics you're going to see with the margin aligned tool set. And margin caller. This is finally the margin aligned dedicated variant caller devised by this group. This outputs a VCF. This is going to be giving you your SNPs. So if you want to look at SNP data on Oxford Nanopore without dealing with phylogenetic trees and probabilistic mapping like we've seen other groups have used for SNP utilization on Oxford Nanopore, this is currently where you want to go. This is the top of the line, I suppose, for SNP calling on Oxford Nanopore data. Certainly there's a lot more that can be done. They say SNP calling is significantly improved after the EM function. What they're showing here is they're showing they've created a synthetic genome. They have a regular genome. They introduced 1% of miscalls or SNPs in that genome, and they tried to see if they could find them. So what they did was, and let me clarify, the SNPs are in the reference, so they changed the reference and sequenced the actual data. That's where the SNPs are coming from. So without the expectation maximization, that's these dotted lines. So this is just the tuned call. So the sensitivity and specificity is not great. With lots and lots of coverage, we can push that out. This is where we want to be, but even down here, it's not very good, okay? You apply that EM function, and now we're very, very sensitive and specific, okay? So we're predicting most real SNPs, and most of the SNPs that are predicted are predicted, okay? So we're capturing all of them, and we have very few wrong SNPs, is what I'm trying to say. This is at 1% substitution frequency. You increase the substitution frequency and things perform less and more deviation, more stuff here. But even then, the EM is pushing out. It has a better performance than the tuned values. And that's because many of the alignments have a high probability of being wrong, I suppose. So 90% chance not of being wrong. It's the 10% chance of being wrong, but it's still fairly significant. So they want to improve that with the EM algorithm. High coverage is better. I mean, that's just the reality of the Oxford Nanopore data. The higher coverage you have, the more consensus you have, the better you're going to call the SNPs. Their performance is less accurate at under 30, but over 60 is when their performance is better. All right, so they want to move to signal-based analysis for alignment, variant calling, and base modification. So if you recall that event align from the last tool set, margin align is working on a similar function, looking at the signal level alignment. So not just the K-mer alignment, but going even more raw to the actual values expected. So aligning based on numbers. Anyone who might know solid, which you may not, in color space, I always think of this the same way. This is squiggle space alignment. What time is it? It's time for me to stop talking. All right, the end. Okay. I'm going to do one thing here. We'll just pop back over to our sequencer so you can kind of see what's going on. Look, we have 60, 70, 50. It was in 70 a second ago in strand. This is going to be changing a lot because there's short reads. Let's see what we've got. Not that page. And look, so we've got a couple, these are going to be artifacts. There's no 35,000 base pair fragment in this sample. It's not there. This is an artifact. We've gotten the 324 channels have gotten data. So actually a little over 50% in terms of used channels. And unsurprisingly, most of our reads are small, and we've generated 14,000 independent reads. So plugging along nicely, nice number of pores in strands, so hopefully we'll let this look pretty good. So I'm actually going to leave this computer up and running for the next 2 days. You may have a chance to take a look at it. It's actually real data for real science, so you can kind of take a peek, see what's going on. Please don't turn it off or unplug it or anything. But you guys can get an idea of what it looks like over the course of a run. So if you have any questions, I am here. Please, whenever you want to know, just let's ask, and we can go from there.
So I'm just noticing that there's like a remote desktop connection.
Mm-hmm.
So you're running the on this laptop.
Yes.
All the data is running.
The data is not coming here, actually. The data is all on that laptop. I set it up just so I can show you the screen. So yeah, I think I can port data from one to the other, but there is a speed function. That's why you have to have USB 3.0. And if it's porting slow, then it's not useful.
So you can't run them on one computer and have it just shoot data to.
Not as far as I know. I mean, you can't upload the data live, but you're still preserving. It's not going away.
Okay.
Yes.
How does your 35,000 base pair read get there?
It's not a real read. It's artifact. It's probably what's happened is some sort of contamination, a piece of dust, something is inside of a pore, and it's causing a blockade in that pore, which causes some sort of voltage change that looks enough like a Kamer that's just, it keeps calling something. So chances are, if I were to find out which pore produced that long read, and you actually can sometimes do that. So if you actually hover over these pores, it tells you what the length of the read last generated was. So if you find that 35,000 pore, you can probably reverse the voltage and unblock it, but you'll probably find there's something wrong with that pore. It's definitely not a real read.
Yes, it's manually.
So you have to do it manually. Right now, yes. I mean, there is a process where it unblocks part of the script, so that does happen, but it doesn't do it for a very long period of time. If you want to do it longer than the script does, then you'll have to do it by hand. I have found that some pores, just nothing you can do gets them unblocked. It's just, that's the way it is. If you go back and some of these artifact reads and look at them, you'll see a normal read, normal read, normal read, then 10,000 bases that are like a 5-mer repeat over and over and over again. So, yes?
Where are the different groups that participate in the MARC located?
Okay, so there's the labs that participate in the MARC there's one here, that's us. UCSC, there's one in the Netherlands, there's one in the two in the UK. 1, 2, 3, 4, 5. There's one more. Where is the other one? I think there's 3 in the US and 3 in the UK.
So here, you said UCS?
UCSC, yeah.
And
Yeah, and so one University of Oxford. There's ZF Screens in the Netherlands. I know I'm missing one. I can't think of where. University of East Anglica is one of them. There's one more, I think. So we unfortunately don't have a lot of folks in South America, Africa, or New Zealand, Australia involved in MARC at all. We do have a couple in New Zealand, but none, I think, in China or Asia at all. So around the world as much as possible. Yes?
How much does it cost from sample preparation to the old chip data, all that stuff? How much does it cost? Just curious.
Well, so the reagents themselves, the actual reagents, it's $500 for the box of reagents. I think maybe it's $599 now. That's good for 3 runs. Each flow cell runs about $1,000. Okay, so that's about $1,500 for the run. The tools, fortunately, these are all freeware, so you don't need to pay for any of these tools. The computer, if you don't have one, the computer runs approximately $1,500, $1,000 or so to get a computer capable of running this. You may have one already, and maybe people who can get it cheaper than I can. So it's a little bit of investment. It's not the cheapest sequencing platform, but it's nice because it's very portable, it's very small, and then they have a lot of rapid and fast. And we've seen vast improvement of the instrument over the last 2 years, and we hope to see more. Any other questions? I can keep you here longer. I can show you more videos. All right, well, y'all are free to go or ask questions and hang out. So down a little bit early, but yeah, not too much. Enjoy your lunch.